From Microbe TV, this is Office Hours for Wednesday, the 26th of February, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet, where you'll get facts about viruses and maybe a few other science things as well. Tell us where you're from, and while I'm waiting for that, let me thank our moderators who are here tonight. We have Les from California. We have Steph SF from San Francisco. Thank you for joining us. Tom Steinberg from Western Oregon, and Vanity Nutrition from New York. Thank you for joining us. It's becoming increasingly necessary to have moderators. They're just people who jump in and say dumb things, and we don't need it. What's the point? It's getting more and more dumber and dumber is this country getting, unfortunately. So what's up for tonight? I want to talk about, oh my gosh. I want to talk about the measles outbreak in Texas. I want to talk about what's going on in Africa. Some weird outbreak that, again, nobody knows what it is. What do we got? 53 dead. Mystery illness. We'll talk a little bit about that. We'll have to talk about RFK. And we will have a mini lecture. I want to talk to you about DNA replication. I know there are other topics that people want. I will get to those. I want to give you a good foundation in molecular biology before we get started here. You know, someone said early on, before the stream started, when reporters asked about the outbreak, Kennedy said there's always outbreaks. What a BS artist. If kids are vaccinated, there are no outbreaks. We eliminated measles in the U.S. by vaccination, and it's only thanks to Andrew Wakefield and his ilk that parents stopped vaccinating and we had outbreaks. So no, RFK Jr., there aren't always outbreaks. And I hate stupidity. I hate ignorance. And he is the epitome of them. By the way, here's a thought for you. I got a bunch of emails from people who said, what's the plan if TWIV gets shut down? You know, I don't have a plan, but I think it's maybe not a bad idea to have one. Someone suggested collecting emails so that I could point people, so I could still post content on a private server and send people links to that and so forth. I don't want to collect email, though. That's just a pain. There are a lot of people on Discord, but not everybody's there. I guess here I could, well, this is YouTube, so, you know, maybe they'll say you can't be on YouTube anymore, because Google, I feel, is just pro bad things. I'm sorry. That's my feeling. So I don't know what to do. Maybe you have some suggestions. All right, let's see who we have here. Elena is in Coxsackie, New York. Yeah, today it got a little warm. Yesterday too. But it's going to get cold this weekend. Here it was a high of 13C and a low of 6C. So, but this weekend it's going to freeze and then it'll get warm again. Here's that quote from Gwen. When reporters asked about the outbreak, Kennedy said there's always outbreaks. What a BS answer. If you were in a qualifying exam or a thesis defense, you'd fail. I'd make sure you failed and had to go back for another year and study. It's disgusting to say something like that. There's always outbreaks. No. If you vaccinate everyone, there aren't outbreaks. Most of Texas is vaccinated. The outbreak is in people who don't get vaccinated, the idiots. And I'm sorry, it's a religious reason. No, I don't buy that. As Poloff has said, there's no religion where it's written in their whatever, their documents, that you can't vaccinate. He said there's one obscure religion. Religions are about taking care of people, not letting them die from not being vaccinated. Idiots. Sorry, folks. We'll talk more about that, I'm sure. Lise is in Columbus, Ohio. Patty is in Massachusetts. Rita's in Idaho, I think. Someone wants to know if you can provide a link to the Cold Bear episode with Dickson. I'm pretty sure it's on YouTube, but maybe someone has it. Ubalubi, Little Rock in the 70s. Les says, I've never expected to see infectious disease progress getting reversed. I've always been an optimist. Yeah, me too. This is ridiculous. There's zero reason. The only reason to cut funding is to enrich wealthy people because, they have to make a tax cut and they have to come up with the income or whatever. It's disgusting. It is absolutely disgusting. As Gandhi said, despots always fall. Always. Yeah, you just may screw a lot of things up until you get them to fall. Brian is in Montreal, -1C. End route. Tired and sad. Yeah, I know what you mean. But we can't give up. You got to take your tiredness and sadness and turn it into energy. I give a lot of money. I write letters. I do these podcasts. Find something to do. Go to meetings. Wayne is in Urbana, 48F, 9C. Will is in Changsha, 10 degrees C and cloudy. As I said, Tom is in Western Oregon. There was an interesting question here. Where was it? I think it's gone away. Basically, Elena asked if I would ever have RFK Jr. on TWIV. And the answer is never. TWIV is for scientists. He's not a scientist. He lies about science. He gets it wrong. And he doesn't correct it. Scientists tell him he's wrong. That's not a scientist. So he would never, ever, ever be on TWIV. Would do no good. We could argue with him till the cows come home. He'd still stick to his beliefs, and he would say, yeah, I went on TWIV and I whipped those guys and gals who think they know all about viruses. I whipped them. Yeah, that's what we need. Tatiana from Birmingham, it's 67F19C. Jem is in Hampton, VA, where it's 53F. Now, here's an interesting comment from Will in Changsha. Feeling very angry today at the death of a child from measles caused by old order Mennonites not vaxing their kids. P.S. I'm a Mennonite. So tell me, Will, is it written somewhere in the codices of Mennonitism that you can't vaccinate your kids? And if so, what is the reason? And are they happy to have their child die? What is this as a sacrifice to the Mennonites? What is this? It doesn't make any sense. And there's going to be more. Mike and Lori, sunny in Seattle, 12 degrees Celsius. Kang is in Chicago, 4C and cloudy, missing Dickson. Yeah, Dickson. We did a nice tribute for Dickson on TWIV last Friday. I hope you checked it out. Everybody was there. And we all talked about him. Patricia, RFK Jr. is a personal injury lawyer. He consults for firms that raise suits against companies that manufacture vaccines. He makes millions of dollars a year doing that. At his confirmation hearing, Elizabeth Warren asked him, would you be willing, that's a conflict of interest, she said, would you be willing to sacrifice that income if you're confirmed? He said, no, I'm not willing. And then the next day he said, I'll give it to my son, which is not much better, right? Ay, ay, ay. Does he have a single neuron? Dagfinn is in Oslo, 2 a.m., 2C. Leo is in Mendocino, 13C. Dean is in Wisconsin, 44F. Margot is in Phoenix Metro, 90. Wow, that's very hot. Yeah, here's the question. Would you ever... Nope, 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 nope. By the way, Aylin is in Westchester, New York. Thoughts on the new USDA policy on avian influenza? Yeah, there's a lot. They're going to invest up to $1 billion to combat avian flu and reduce egg prices. So they're going to instill biosecurity measures for all poultry producers. They say the 150 facilities that follow these protocols have only had one outbreak. So that's not bad. They're going to have epidemiologists trying to trace outbreaks early on. USDA is going to pay for this. They're going to pay for farmers whose flocks have been decimated or destroyed. Remove regulatory burdens on the chicken and egg industry. Explore pathways for vaccines and therapeutics. Consider temporary import-export options to reduce costs. Hey, did you hear we're getting eggs from Turkey? That would make them turkey eggs, right? So I think this is okay. The problem with vaccination is it doesn't prevent transmission. It prevents disease. So the virus is still going to spread. And it will protect your flock. You won't have to kill them, presumably. But I don't know how other people are going to feel about buying your chickens under those conditions, right? Jill, hello, welcome from Delaware, if I remember correctly. Carol, our favorite nephrologist from Bakersfield, where it is 24C. Lovely. Maureen is in Northeast Ohio. Laura is in Washington, PA. Anatomy and physiology tutor is in Vancouver with 51F. Joseph is in Ontario. Tanvir is in Louisiana. Margot is in La Grange, Illinois. Michael is in Wales, UK. Okay, Rita's confirmed. Southwest Idaho, 49F. Eliza, Southern Ontario, 4 and clear. Tracy's in Chapel Hill, 12 degrees Celsius. Jeff is in Elgin, Illinois. Jonathan's in Auburn, Alabama. Elizabeth is in West Virginia. EHS Talent is in Eastern Pennsylvania. March 716 in Central Virginia, 11C. Jill, 60F in Wilmington. What is disc golf? Tell me what that is. Jefferson Space is in Miami. Read to vanquish the dumb, indeed. This would be the dumbest administration ever. SRR is in Paso Robles. Steve. Logotic is in Chicago, 4 degrees C. Pete is in London, plus 4C at 1 a.m. Gonna get a tooth fixed. Tell me about this ridiculous stuff, Lucky Sixes. This is the one with the subway. Oh my God. I don't understand what's happening. I mean, it gets worse and worse. Yeah, I know I was going to do Twitch here, but it turns out that, oh, actually, yeah, I'm sorry I didn't do it. I'll do it next time. Rona on the Long Island. It's Tona, but Rona, whatever. Harry ozone, not a miasma here in Asheville, where it's cloudy in 56. Yeah. What is the miasma quote from TWIV? You can buy a T-shirt that says it. You clear up the miasma of something. Oh my gosh, someone must remember. Cheryl is in Santa Rosa, 70F and sunny. Okay, Patricia wants to know, why would you get shut down? Because they don't like truth. Isn't that the trend these days, that they're censoring truth? And YouTube is part of Google, which is a big company, which is full of, populated with billionaires who just want to get richer. So that's what but people are writing me and asking me, what would you do? What's your plan if TWIVs get shut down? Okay. Will we be able to contact the TWIV team? Well, yeah, of course. First of all, well, I could always stream somewhere else, right? And you're going to find me, but you have my email, vincent@microbe.tv. And Discord, if it got shut down, I would do draconian things to get in touch with all of you. And we'd find an alternative place, right? So Sally says Blue Sky. Yeah, well, Blue Sky is okay to put links and stuff in discussions, but you can't have an hour, 2 hour podcast there. So you'd have to link to it. But we could reconvene on Blue Sky, yeah. Marge will follow TWIV underground. Well, I'll tell you, folks, I'm not changing my ways. I'm still going to be critical and mad and tell you real science. That's what I'm trained for my whole life. And no idiots who don't know any science is going to tell me what to do. It's not easy to know science. And especially if you've never taken any science courses, which, this man who's head of HHS has never done. Charles in Madison. 4C and overcast. Not so bad today. Angry Penguin is north of Toronto, where it's -2C. Hello, Hans from Guayaquil, Ecuador. 30 degrees C. Holy cow, that's hot. It's really hot. Julie S. is in Hughesville, PA, 45S with rain. Donna, I tried to post a comment on the immunology lecture. Yeah, the commenting isn't working. A lot of people have told me that. I do have website people to say, this is broken, will you fix it? So I will do that. Yeah. So Uba Luby loves the Discord, and you get my excuse me, you get notifications for videos, but of course, if they're shut down, they're all on YouTube. By the way, Vimeo, I could put videos on Vimeo. They seem to be agnostic; they don't care. So Vimeo, I do have a channel on Vimeo. It doesn't have much there, but if things go down, let me find the channel. Let's see, Vimeo, TWIV. Vimeo, yeah. 13 years ago. Oh my gosh, this particular this particular video. I'm just getting you the. Now I can't log in because the password is not matching, but there are quite a few videos there. Why can't I find? And they're old. They're all very old. How do I get, like, is it microbe TV? Maybe. Let's look. Would I have had the sense to do that back then? Nope. Anyway, maybe someone could find the Vimeo channel for microbe TV. And that's where I'll put videos because I can make a video and post it there. It doesn't have to come from YouTube, so that would be good. Okay, there's the solution. Pat is in Nova Scotia, where it's 2C. John, 48F, 9C. In Minneapolis, we consider this a heat wave for fall spring. It's not Sally's view of religion. No. Lori is in Tomball, Texas. Visto is in Sydney, maybe 37C later. Frank is in Santa Barbara, 70s. Elizabeth says if YouTube started censoring that bad, there would be a lot of people affected. Another platform would get started or expanded. Yeah, maybe. Maybe. DH 70s today in Atlanta. Mr. Doreen, Elgin, Illinois, 10C today, plus 10C. Patricia's in Massachusetts. David, I think I listened to 4 hours of TWIV today while doing work at home. That's really cool that you would do that. Thank you so much. I hope you learned. GSD, Jonathan, 17C, and Encinitas. Influenza vaccine planning suspended. Well, I don't know about that because it's not a U.S. thing. It's a WHO thing. So the rest of the world is still going to plan what the next vaccine should be. Tell me what you're thinking there, Visto. Scott is in Seattle, 12C. Tom, sufficient unto the day are the evils thereof, which means don't borrow trouble. Is there a credible threat of TWIV shutdown? No, people have just asked me, what's the plan if it does get shut down? And I didn't have one, but we have Discord and we have Vimeo. And something else will pop up, as someone just said. So, HZoo, hello. Austin, Texas, 73F. Anatomy and physiology tutors in Vancouver, where it's 12C. The last infectious disease pandemic cost $13 trillion. Yeah, this next one will be even more. Paul is in Longmont, Colorado, 7C. David says, is there a scenario in which measles could get out of control? I don't know the answer to that because the overall measles immunization rates in the U.S. are pretty high. In fact, in Texas, outside of this religious community, it's 97%, which is really good. Now, let's say that it was 97% across the U.S. I don't know if that's correct. I'd have to look it up. But that still means there's 3% people who are not immune. But also, everyone who gets immunized does not develop antibodies. There's I forget what the number is. 5% of people who get measles vaccine do not make an immune response. So there are going to be people who are not immune. And if the jerks who don't get vaccinated become sufficiently numerous and have outbreaks, that's going to spread into the communities where people are well vaccinated, but there are 5, 6, 7% who are not. And yeah, but never, I don't think out of control, but lots of cases because we can go in and immunize if we're allowed to do that. If idiots don't step in and say, no, that's a bad vaccine. You know, at some point, people need to stand up and say, F you. You don't know what you're talking about. What's the story? You can't let people die. Jesus. I got to relax. Oh my gosh. Kip, nice to see you. Thank you for your support of science communication. Well, I think there have there been 1,000 yet total in the U.S.? Yeah, it's close. Yeah, it's disgusting. DH got 3 responses from senators. Keep writing, folks. Look, write every week and tell them different things like this outbreak in Texas. I don't know what you do with a religious community who won't get vaccinated, who won't vaccinate their kids. I really don't know what to do about that. I mean, it's just crazy, right? I mean, if they have more deaths in their community, are they going to change their mind? No, this is not religion, okay? This is being evil because religion is taking care of your kids, and that includes vaccinating them. Jesus. Oi, right? Keep writing, folks. Hello, Abdur Rahman in Brooklyn 11C. H2 is worried about measles in Texas. It's in a couple of colleges. It will find the non-immune people. There will be more. Yeah. The tragic measles death was a certainty. I expect the casualties will be downplayed or something else until red states can't hide them. Yeah, RFK always has already said there are always outbreaks, which is a flat-out lie. As I said, if you are vaccinated, there are no outbreaks. The reason they had an outbreak in Samoa is because they weren't vaccinated. As soon as they vaccinated, the outbreak stopped. Can you put 2 and 2 together? Apparently not. This is the latest requirement for getting a confirmation in the Senate to not be able to put 2 and 2 together. Lewis said from Bay City, Michigan, 36F, watched the latest clinical update. Heard y'all talk about H5N1 in cats in Michigan District Health Department 2. I work with them. Small world. Yeah, it is a small world. It is. Thank you for your support of science communication, H. Shu. Really appreciate it. You ought to go see Rich Condit sometime. You ought to have lunch with him. You're in Austin, you know? It's close. Hello, Mark Martin. Back in Tacoma. Mark visited the incubator on Monday. We had lunch. We took a picture. Mark came to my virology class. I asked him questions. He was very good. He got all the questions right, all the quiz questions. Nice to see you, Mark Martin. Mark, the phage doesn't dance anymore. Where's the battery I have to replace? Is it in the actual phage or is it in here? Because there's a battery in here too. Let me know, please. It didn't last very long. Pete says the equivalence to religious child sacrifice of not vaccinating is an interesting point. Will says non-vaccinating has nothing to do with faith. The problem with old order Mennonites is that they reject modern science. Why? They exist because of modern science. Anything they do is a result of modern science. So they're just being inconsistent. Elizabeth says the TWIV tribute to Dickson was really wonderful. It was very nice, spontaneous. And by the way, what was I going to say? The letter. We had a lot of letters, which we didn't have time to read, but I posted them. So if you go to the letters link in the blog post, you'll see them. It's really nice, very nice. And they still continue to come in, of course. Marge says it was nice out today, so I started working on my vertical garden. Thank you, Dr. Despommier. Lori says the Dickson tribute was wonderful. Scott says, I'm sure there's a religion for breakfast video about the various vaccination, medication, blood transfusion strictures. Yeah, after the fact. But when the religion was founded, they didn't have in it, you can't have vaccines. Marx says, sadly, we have dealt with deeply religious people who will not accept blood transfusions. Heartbreaking. The question is, when it comes to a kid, right, withholding vaccines and blood, that just doesn't seem right. I don't know about that. So the adults, you can do what you want. You can die if you want, sure. But the kid doesn't know any better. It's not fair to screw the kid over because of your messed up beliefs. I'm sorry. You can have whatever religion you want, but no transfusion, no vaccines, no science. Give me a break. What are you dealing with here? Anything else is fine. Worship a goat, whatever. But it's ridiculous. Unbelievable. So, JT, I got my measles vaccine in 65-ish. Should I get a booster? Well, I cannot give you medical advice, but Daniel can. And we have a bunch of these questions from people that are going to get answered tomorrow on the clinical update that we'll release on Saturday. All right. I don't know what he'll say. I got measles vaccine in the 60s also. I'm not getting a booster. I haven't done any antibody titers. Just not getting a booster. I think the immunity is probably quite good. But let's see what Daniel says, okay? In memoriam, Dickson Despommier, thank you. Carol loved the tributes, the story of him kissing every fish and releasing them. Yeah, I saw him do that, man. I got it on film. I have to put that video up. Mark is in Maui. 25C. Harry Ozone, maybe he was missing from the Dickson Memorial. You're right. You're right. That's a great point. We got this together really quickly, so I'm not making any excuses, but she would have needed permission to join. I don't know how long that takes. Rita says, not H5. The conventional flu is showing up with encephalitis, encephalopathy in a few children. I think that happens, if I'm not mistaken. Eliza just finished Dr. Offit's book, Do You Believe in Magic? About alternative therapies. Great read. Really? There's a book called Do You Believe in Magic? I didn't know that. You're right. Do You Believe in Magic? The Sense and Nonsense of Alternative Medicine by Paul Offit, published in 2013. It's quite an old book, but that's okay, right? Thank you for mentioning that. Appreciate it. No, he has no neurons. The bear worm ate them all. Thanks. Tom says, thanks to comments here last week, I applied for and received COVID test kits from the U.S. government via the post office. I'm amazed, Tom, that this is still ongoing. I'm really amazed. Pete knows someone in the UK whose partner is profoundly deaf due to childhood measles. Yes, deafness is another side effect. Yes. Julie, listen to Daniel tomorrow. Okay, he's going to answer this question. The 50s and 60-somethings, do you need a booster? And it is more relevant now because there's a lot of measles in the U.S., right? I totally get that. The chickens get chicken pox. No, so chicken pox, right, is caused by varicella virus, which is a human virus. Chickens do get herpes viruses, but not chicken pox. Nope. Oh, yes. So I also had measles as a child, and I also got vaccinated. So I think that's why I'm thinking I don't need a booster because I've had the double thing, right? The vaccination and the infection. Rita, health finches get a pox. The silent runner tells me Mennonites are not a monolith, quite the opposite. There are many small sects. Some are conservative, some are liberal. Okay. I'm not trying to single out anyone in particular. I just don't understand why you wouldn't protect your children. I don't get it. Religion is supposed to be love and caring for other people. So it doesn't make any sense to me. But that's it. No more of that. Who's going to want to work for the USDA when you could get fired? I don't know. They're just putting out all this big web page on what they're going to do with bird flu. I don't know if they're going to implement any of it. Who knows, right? Used to said, are the anti-vax Mennonites a minority splinter of the religion? I don't know. Maybe someone else knows. Seems there's some people knowledgeable about Mennonites. JT is in Porto, Portugal. Welcome. John is in northern Thailand. Sorry. Abdul Rahman, what are the odds of avian flu mutating enough to spread to humans? Is culling the flocks enough? Do we know for sure the dead birds won't be sold on the market? Okay, so the culling of the flocks is actually, it is meant to stop onward transmission, but it's also a humane thing because the birds are all going to die anyway, pretty much. 90% of the flock is going to die. And instead of letting them suffer, you cull them, right? No, it's not enough. The virus continues to spread. Wild birds spread it left and right. We're not culling wild birds. We can't. So what are the odds for it mutating? Mutates continuously, right? It reassorts. So the odds are pretty good. But it's been doing this for a long time, since the 90s, and nothing's happened. Now, since 2020, we've had this global spread, off across the Atlantic and North America and so forth. So that adds to the mutation and reassortment. And every new animal infected adds to it as well. I don't know what the odds are. I think they're pretty good, but we see. I personally think that transmission is not a fit situation for this virus. I think it's very fit where it is now and not transmitting, and I'm not sure that those mutations are going to arise, but that's just my guess. But there's plenty of raw material for mutation and reassortment, that's for sure. Yeah. Thank you for your contribution to science communication, Questella. A coffee cup with the words tired. I'm not tired. I'm a little frustrated and disappointed. I'm really disappointed in the voters of this country, especially the ones who didn't vote. I'm disappointed in people who'd rather enrich themselves and their friends than take care of the world. I was always so proud of the U.S. They took care of the world. We had the financial means to do so, and we did it. I don't know what's wrong with that. And to make a 360 on Ukraine, this seems ridiculous. There's no reason for that. Really? You think Ukraine invaded Russia? What are you, out of your mind? Every Republican knows that's a lie. I can't stand the prevarication. There's another word, prevarication. Does anyone know what that means? Ugh. Eggs from turkey should make splendid shakshuka. I don't know what that is. Explain. Disc golf is frisbees. Okay. I didn't know that. Jay Shuri is, wherever he is or she is, it's 50 degrees. Kang says, is there irony that this administration will pay for epidemiologists for chickens, but not for children? Let's see if they do, Kang. It may be all just writing on a website, right? Well, they made a point to say early on that the Democrats didn't do any of this, and we're doing it now, right? Ian says, aren't most fresh and frozen chickens awash with salmonella and E. coli? Yeah, of course. That's why when you cut chickens raw, before you cook them, you got to be really careful about cleaning the board, the knife, your hands, and so forth. Otherwise, and the thing is, they not only have E. coli and salmonella, but they have drug-resistant variants, which you could get, and then many years later, if you have surgery, they could rise up to hurt you badly. They replace your own E. coli with drug-resistant versions. Yep. Can we develop a strain of chicken that's resistant to avian influenza through selective breeding? I'm sure people are doing that. There's so many ways that you could do that. And what you would do is knock out some gene in the chicken that's essential for influenza virus reproduction, right? It could, but the thing is, it can't be essential for the chicken. Then it won't live, right? And so there are a number of host proteins that you could do that with. Sialic acid, which is the receptor, I don't think you can get rid of that. I think that's too important. And it's everywhere. Breeding, I think that's hard because you'd have to do a lot of breeding. You'd have to infect at a low dose, get your survivors, infect them again, on and on. I think it's better to do some genetic engineering of the chicken. Then it would be genetically modified chicken. I have no problem eating it, but a lot of people wouldn't. Matri wants to know why two lineages matter. Why couldn't one lineage have been leaked to the wet market and the other evolve from there? It didn't. It didn't evolve from the first. They're two independent lineages that are not one evolved from the other, and they were leaked independently from animals into the market. So the people who say it came from a lab, well, a person could come from the lab and bring one lineage, and then another person could go and bring another. The odds of this are really small and highly unlikely. And that would posit that there are two lineages in the lab as well, which doesn't make any sense because the lineages come from independent animals that were brought into the market already infected. Okay? The two lineages is huge. It completely destroys lab leak hypothesis. And that's why they never mention it, because it completely destroys it, right? And it turns out that there probably were more than two spillovers. The others didn't take off. When you do the predictions, you can see that. Sharon from Tasmania, Midsummer 15C. Wow. So it is summer down there. Yeah. I was having a discussion with someone the other day. Yeah, because it's winter here, right? Michael says we're going to have access to updated flu and updated COVID shots next fall. That's a really good question. There are committees at FDA and CDC that review the data for new vaccines, and I understand that they're messing with those committees. Maybe they're going to load them up with anti-vaxxers, and so you won't have any new vaccines. So I don't know, Michael. I would say in a normal world you would, but this is not a normal world anymore. Sally says Costco had quail eggs. They were pretty, but I was afraid of them. All right. Doreen says Lagrange is pronounced with a long Lagrange. Okay. Also, the Willis Tower in downtown Chicago is pronounced Sears. Okay, thank you. Okay, Mark's thought is finished before people get wise again. What was the Oh, I don't know what your other thing was, but Visto, a miasma of anti-think. That's TWIV cuts through a miasma of anti-think. And we have that on a t-shirt because a listener wrote us an email and said, I love TWIV. You cut through the miasma of anti-think. It was during the pandemic, so we made a t-shirt out of it. Miasma of lies and scams. Yeah, that's what it is. Yes, Costello will find me because she found me at the first stream. She found me and she's been here ever since. The Blue Sky needs to have a way to have live chats. Yeah. We will find you. I like that. Well, I'm here in the Northeast, New Jersey, New York. The incubator is on 7th Avenue. Vincent at microbe.tv. What else? We got the Vimeo channel. We got, well, the WordPress sites. Those are not Google, YouTube. Those are independent, so you can still find stuff there. So I think we'll be okay. They can't stop us from finding each other through the internet back alleys. Indeed. It says if YouTube becomes unhospitable, we will follow you anywhere from Blue Sky to 2 cans and wire. Can that be great? 2 cans and a wire stretched between them. Why no love for Twitch? I just forgot. I'm sorry. Someone told me I should stream to TikTok, but you can't from this software that I'm using here. But let's see if we can stream to. Well, I can't. I don't want to mess it up. I'm pretty sure we can stream to Twitch. It's a matter of me remembering, Quistella, to do that. I was on Twitch twice. You know that. I like Twitch. JT says, aren't there 2 types of measles? One not too bad, the other can kill. No, I'm not aware of multiple strains of measles that are like that. No. I could be wrong, but I'm not. It doesn't pop right into my head, which virus things tend to do. Mr. Doreen, wherever you will go, we will follow. Thank you. You're very kind. Others will be shut down first that are much more political. Perhaps. I think the shutdown of science is simply to get the money, right, for other purposes. El Mug is in Pueblo, Colorado, 8C. Abdul Rahman says, if we start a podcast that has a political bent, would you like to come on to provide a scientific view? Yes. Thomas says, countering a miasma of anti-think. There it is. That's the t-shirt, countering a miasma of anti-think. That's appropriate now, right? You ought to go and buy your t-shirts, countering a miasma of anti-think. It's cafepress.com/twiv. I think I have one, but it's too small. I have to get a bigger one. There's a lot of anti-think going on right now, as there was during the pandemic. Thank you, Tom, for finding that. Rona is my real name. I'm sorry. Tonaru. I'm sorry, Rona. My apologies. This is very funny. Where does this come from? Vincent is the sodium, we are the water. That's very good. I like that. Webinars through Zoom. God, I hate to go back to Zoom. It's such a sucky platform. But if necessary, but I think there are other platforms we can use. Yeah, you can. Those of you who donate on Patreon, you can find me there. PayPal as well. Plenty of places to find me. Pete, Google demonetizes YouTube it doesn't like rather than banning them, and you aren't monetized. Make sure you're secure from being hacked. I think we are. Who knows? Okie dokie. Vincent, can you explain the anti-inflammatory actions of azithromycin? So they're thought to act on macrophages. Okay. So macrophages make a variety of anti-inflammatory molecules, and azithromycin inhibits their production. Etoi 2001. Gosh, I'm looking at that and trying to parse how to pronounce it. Iotoi, Cincinnati, 15C. Welcome. Jill says disc golf is frisbee, throwing frisbees into baskets. There are about 9,500 disc golf courses in the U.S. It's an excuse to hang out in a park and invent new curse words. How are measles testing centers in Texas being run? If everyone who thinks they might have measles goes to the same place, wouldn't they all get measles? I don't know how they're being run, but they should. Obviously, everybody has to wear a mask, right? And they have to space people out substantially. Maybe they learned something from COVID. Vimeo is known to delete videos, sometimes older ones. Well, I have really old ones. They haven't deleted anything, so they've been okay. But I just put up my lectures and various podcasts. Nothing. But I did put up the Beyond the Noise that was taken down by YouTube, and it was fine. It's still there. Not a problem. Don't announce your password. Don't worry. I don't remember any of them. They're all saved in various programs because they're just, well, they're not pronounceable. On the CBS News this evening, their medical doctor said measles vaccines prevent infection. To my knowledge, there's no data that says it prevents infection. It prevents measles. But a lot of people think it does. It's one of these, what do you call it? When something gets established without any proof, becomes an old wives' tale. I hate to use that expression, but folklore. Same thing with polio. Oh, oral polio vaccine prevents gut infection. No, actually, it doesn't. How do I know this? Well, here's one example. Here's one piece of evidence. When you take oral poliovirus vaccine, you shed it for days to weeks to months. So how can it keep shedding if it's preventing shedding? No, it doesn't, obviously. Ay. Krista is in Western Massachusetts, 35F. Say hello to Alan. Ask the Pool Man is in Long Beach. We're at 66. Nice swimming. Very good. So Wayne says, I don't think you'd travel to a central testing center. That'd be in Texas. You might have to travel to a doctor's office or clinic where they take samples, which are sent to the test center. Okay, Hans says, knocking out PRNP gene, the prion protein gene, doesn't seem to be incompatible with life. So is it possible that such gene therapy could be used to prevent prion diseases? If you're knocking out in mice, they live, but who knows what? Maybe they can't sleep. Maybe they have horrible nightmares. Maybe they have all kinds of things they can't tell us. So I'm not risking it in people. You can't. It's an unethical experiment. Yeah, unfortunately. Gloria is in Sioux City, 48F. Many people have no science knowledge and have to listen to HHS director. I'm worried for everyone. Why don't they listen to us? They can get correct science and banter and ranting as well. Daniel is on this. Well, on the surface of the moon, it's minus 250. Okay. Barb Mack, welcome, our moderator. Good to have you. Crestella listened to 4 hours of TWIV today. Well, I cried about how hateful everyone has become. Then I went and bought wine. Yes. Well, yes, certain people have become more hateful. But I notice in my daily commuting that many people are still nice and courteous. They don't seem to be hateful. But yeah, a lot are. Now they seem to be validated by this horrible administration who is just full of hate, right? How can you call the January 6th uprising a day of love? That's only if you hate people. Unbelievable. John wants a synopsis of the hygiene hypothesis. Well, the hygiene hypothesis said kids should grow up in an environment where they get bacteria to contribute to their microbiome. And studies have shown that kids who are in such environments, outside a lot, not indoors all the time, and not contacting the environment or even other people, are healthier and have fewer diseases. Very hard study to do. Excuse me. It's a observational study, right? And it's full of confounding variables. So I think there's no question that the microbiome is really important, right? But not clear to me that you need to roll in the mud to be healthy the rest of your life. I think you just live normally. You go outside, you walk around, you interact with people, you eat food in restaurants, which has its own microbiome. I think that's fine. I don't think you need to grow up on a farm to be healthy. Is the Uganda WHO page still dark? No. And let's see. That's a good question. Uganda WHO. Well, I don't know what page you're talking about, but what do people think about this mystery Congo disease? 53 dead in the Congo. The people who died apparently died in 2 days, which to me is crazy. 2 days, 48 hours. They think it started with a couple of kids who ate a bat, and they died. They took samples. They tested it for Ebola in Marburg. Nothing. It's not there. So we don't know what it is right now. Remember, this happened last year, right? At the end of the year. Lots of deaths, and it turned out to be a severe form of malaria. I'm not saying this is, but I'm not sure it's anything new. I'm not sure the kids eating the bats means anything. It sounds cool, but really? I mean, did they? Oh, never mind. Will says, being retired, I always listen to TWIV at home. Sometimes I listen as I do my housework, unless I want to take notes. So, how do you find it? Is it hard to understand? Because I think it's very hard. I think we don't explain a lot of things. I try. Tess is in Canton, MA 39F. Dr. Dre fan would love me to talk about bacteriophages and how to isolate them. Oh, we do from time to time bacteriophage episodes on TWIV. As you know, bacteriophages infect bacteria. So you could identify phages that infect just about any bacteria. And there's a great program for high school students called C-phages across the U.S. where they go in their backyard and they get some dirt and they bring it into a lab and look for phages that infect a particular bacterium, a mycobacterium, a non-pathogenic mycobacterium. They take extracts of the soil and they mix it with the bacterium and plate it on agar. It's really easy to do. The question is, what bacteria do you want to use? And that's what you should figure out first. I mean, there are lots of bacteria that you can get out of your mouth or from the dirt or from trees or plants or whatever. And then you could take soil. For example, soil is a good source. There are lots of phages in soil. And you suspend the soil in some saline, let the soil settle out, take the saline, mix it with the bacteria, and then plate it out and with an agar overlay. You'll find plenty of phages there, but also other places. You can find them in your gut. Plenty of phages there. You can find them in water. So, yeah, it's a little bit. Visto, Paul Offit on CNN. March meeting of outside advisors to the US FDA to discuss composition of flu vaccines has been canceled. Right. So I'm thinking, Visto, of the bigger issue, which is which vaccine to use. The FDA takes the recommendation of the WHO committee, and the WHO committee is still meeting. So, that recommendation will still be there. Maybe manufacturers will just take that. I just don't know how it's going to work out. I'll ask Paul. We're doing Beyond the Noise on Monday. What do you think of the zero COVID movement and those recommending that social distancing and masking continue? I don't think it's realistic because we now have ways to minimize the severity of COVID, right? We have vaccines and we have antivirals. And don't tell me that they don't work. Just stop, all right? I'm the virologist, not you. They work. And social distancing and masking is abnormal. And you're going to get social issues if you continue to do that. That's why people don't want to do it normally. They want to be near people and they don't want to mask for various reasons. So I don't think it's needed at all. Now, if you are immunosuppressed severely and constantly, sure, mask and distance. But not everybody has to do that. Eliza B., I listen to TWIV in my car on my way to visit my mom. She's a chemist, 96. I share the bullet points with her each week. That is just great, Eliza. I love it. It's so cool that she wants to know the bullet points. Does she like the bullet points? Or does she not care? Rita says vaccination rates in Ohio are very low. This is what, for MMR or all vaccines or what? Tell me, Rita. Lovely Rita, meter maid. Oh, that's a great song. So don't get offended, okay? Jason says, well, maybe the real question is how many children will be born in the next few years that are unvaccinated. I think most parents understand the value of vaccination. There are pockets, as you see, in Texas, and they're all over the U.S. The issue is if they grow, right? If this guy, RFK, influences people into thinking that vaccines are bad. This is horrible because he's full of misinformation, of course, and no one should listen to him. So we have to figure out how to counter him. But as soon as he starts saying things that are wrong, I will make a short 1-minute video saying, this is wrong and this is why. That will be my contribution to countering his nonsense. Ian wants to know what % of people don't respond to Hep B. I don't know. Let's look. All right. 5 to 15%. Yeah, so you could easily be in there. Yeah. David A. says, Jason Stanley, an American philosopher, says that university presidents need to start speaking out, as universities are an essential part of democratic institutions and education is under attack. Yeah, they need to speak out, but they won't. They don't want to disturb their funding, their precious, bloody funding, which is their single-minded thing that they got their funding and there's no other way to raise money for education. No, not being creative whatsoever. But yeah. Oh, there was a great in the pipeline. Did you see where two corporate CEOs are at a meeting in D.C. and they're talking about how they can work with the current administration? They're basically BSing because they don't want to insult the current administration and spewing nonsense. And as he said, Derek Lowe, it would have been better if they kept their mouths shut. I love it. You should read it just for that. Love it. In the pipeline. Jason said maybe someone should pay people to get vaccinated. Well, you're not going to pay the Mennonites, right? Because they don't think it's right to get vaccinated. So that doesn't solve that. How will we get vaccine needed for outbreaks of something if they won't make and distribute it? Well, it's not clear that they're not going to make vaccines and distribute them. I mean, they're doing a lot of hot air balloon stuff, but I'm not sure that we're not going to get a new flu vaccine or a new COVID booster, or if there's a pandemic, I'm sure that they will make a vaccine. I don't know who, because he's in the process of dismantling the NIH. Maybe the companies. But and this is the thing about this in the pipeline comment about these two biotech CEOs. And one of them was the CEO of Pfizer, Bourla, Alex Bourla. Academics are where you get the people who work in your friggin' company. And he's in the process of destroying academic science. And you can't say anything negative about that? It's disgusting just so you can curry favor with him. It is disgusting, Mr. Bourla. Oh, my gosh. And Derek Lowe called him out. It was great. It was another CEO, too. Abdul Rahman said, I was just chatting with a couple of local politicians. Even for state senators, 20-plus emails, letters gets attention. Smaller offices, even 5+ is enough to force a response. So I would say every week you should write something about what's going on. Why destroy the NIH, which has been great for science? Why prevent this vaccine committee from meeting? Why prevent the public announcements that are needed to get NIH funded? So you know that Trump cut off funding of NIH grants, right? And then the courts said, you can't do that because Congress has appropriated this money. You cannot just cut it off. So what did he do? He shut down the communications that are needed to give out the money, the Federal Register publication of the meeting and so forth. So no money's coming out because of that. Isn't that insidiously disgusting? It's what a snake would do in a viper's pit. People call me Jerry. Any thoughts on boosting at 60 years old? Boosting for what? Yeah, flu and COVID boosters are recommended at certain times, yeah. But I don't know if you're worried about measles. Then you have to wait for Daniel Griffin's answer. Texas, I understand many have medical exemptions, 14% compared to 3% nationally. Yeah, these medics are not doing anyone any favors. I heard it's even higher than 14%, but let's assume it's 14. The rest of the country, I heard a guy say, a vaccine doctor say, the medical exemption rate should not exceed 1% nationally. And I think there's some counties where it's even higher than that. So it's disgusting. Docs, you went to medical school. You took a Hippocratic oath. Do you remember it? Oh, my God. 148 likes, 267 people are here listening to me rant. Please hit the like button so we attract more people. Okay, yes, there is Ariozone reminds us there's a Stand Up for Science march in D.C. and also in other cities as well. And Les reminds Dr. Dre fan that TWIV episodes with Jolene cover this. And you could write her an email to TWIV if you want to. Stacy catalyzes. Why does measles not reduce in virulence like Zika? Zika does not reduce in virulence. It's population immunity that ameliorates disease. John, I love hearing Mark's voice in Vincent's favorite lecture on RT. I cut most of it out, but I left one in that was louder because if you boost, he's sitting in the audience and the mic is on me. So if you boost his voice, it sounds horrible. But yes, Dr. Martin got every quiz question correct. And he said in the lecture, which is really great, every question is an opportunity to learn. Yes, Joy. It's really, really good. Let's see. Mark says the battery pack is attached to the discophage. Okay, I will take care of that. Oh, do you have a watercolor? I started taking photos. I took one at the incubator and then I stopped, or two. So I'm going to show them to you because I have many more. And I'd like to show them to you. But I'll show you these two. Receiving 2 photos. Okay. He made some really cool watercolors. He had a theme. Now Mark says these were Mark says these are fungi. Yeah, let's shrink it. Okay, so this is one that's hanging at the incubator. So there's his signature, Despommier. And Dickson always told me they were plants. So he made a whole series with this motif. But Mark said it looks like fungi to him. But I love it. I think it's gorgeous. So that's one that I have in the incubator. And then here's another. This one is not hung, unfortunately, yet. But, same motif. This fruiting bodies, let's call them, on stalks. And I have 3 or 4 more. I have 5 more here at home. I will photograph and show you next week. And then I have some other of his artworks. I have a watercolor, which is not a fungus or a plant. And I have some pictures from an exhibition he did at Columbia years ago. I will show you those next week. I meant to do it this week, but I didn't do it. So that's one. Isn't that cool? It's neat. Right. This is Christian Science and oxymoron. Right. Right. We never had all this nonsense and chaos when we had Dickson. Yeah, well, it doesn't, if he were here, he would talk about it, but he couldn't prevent it, unfortunately. Doreen says, I'll bet Governor Waltz would say, it doesn't matter if you're not into science, science is into you. I like that. It's really good. Rita says, goats are excellent. I don't want a goat, that's for sure. I saw it in a video on Instagram where this goat was trying to headbutt a capybara into the water. Just kept butting and butting, and the capybara was like, dude, I'm not going anywhere. Will says unvaccinating is not just about your own kid. You're putting others at risk. I understand. But the greatest liability is you're responsible for your kid. That's an abrogation of your parental responsibility. You should not be allowed to have kids. Karen says the problem still seems to be treating children as property and not individual humans. Well, they're just going by their religious rules, so-called rules, and it doesn't matter if it's their kid or not. I wonder if they would actually like to vaccinate their kids, but they're not allowed to because of management, right? Management. Martin says, are there risks from live measles vaccine to others in the household that are immune compromised? I'm not aware of such risk. Let's take a look. No evidence that measles vaccine is transmissible to immunocompromised people, but they should not receive it, right? Because it's an infectious vaccine. There you go. Lee says, when children were seriously ill in the hospital, needed treatment, the parents refused, I would get a court order to treat. This only happened a couple of times, but created such chaos. Yeah, my gosh. That's really a difficult situation, right? I don't even know how to respond. Where did I hear Tona says that the excuse using religious objection is exaggerated? No, it's not exaggerated. I think the numbers are good. I don't know if Daniel said that or not. I don't know. Offit's book, Heather says, will open your eyes to the amount of money and lack of regulation that exists in the supplement and all things natural business. And you know, this is what makes anti-vaxxers rich. How likely is this outbreak to spread nationwide? It depends whether they go places, these kids, right? It's unlikely that they're, well, if they went to Disneyland, that's how the outbreak got started, right? We didn't have measles here for years after vaccination was instituted. And then after Wakefield did his nonsense in the UK, then there started to be cases of measles there, and then they came to the US. So all the measles viruses that are circulating came from the UK. Thank you, Andrew. Really good. Do you guys want to hear a mini lecture? Because I could spend the next 40 minutes just chatting with your comments here. Dorothy, I think we'll skip a mini lecture. You know, last time I don't think anybody was listening. Maybe 4 people were listening. I sound like a crotchety old professor. Mini lecture, please. Okay, we'll do a mini lecture. Speaking of COVID test kits, do they expire or become less effective with age? They do expire, but it's not clear that they don't work. It's just that that's as long as they've tested them out in the manufacturer, right? So Daniel says you can use them. All right, we do a mini lecture, okay? And then we'll probably get back to some questions because it's mini. Let's find it. Microbe TV, office hours. What's today? 2025 is the year. Office hours 2/26/25. There it is. Make available offline. Yeah, this is the Dropbox Tango. And we're going to play it. And I want to talk about viral DNA replication, folks. I'm still here. What is viral DNA replication? Well, for viruses anyway, that have DNA genomes, they have to make more of them when they infect a cell. So here are some viruses in the Baltimore scheme that have DNA genomes, right? The double-stranded DNA, the single-stranded DNA, and the hepatitis B virus DNAs, which are kind of broken. They have to be replicated to make new progeny, right? Yeah, when our cells divide, we have to make new DNA also. Right? I just saw Tona's comment. Everyone asks questions so he knows we're alert. Now, I don't care if you don't pay attention. Last week, you were all chatting amongst yourselves, and that's fine. That happened when I did a live stream virology course a couple of years ago. I'm just joking. A few people who want to listen will listen. It's fine. All right, so genomes need to be replicated to make new progeny. And the thing that's very interesting is that DNA replication is always delayed after infection because you need to make proteins, at least one, sometimes many proteins, before you can replicate DNA. Now, there are rules of DNA replication. So DNA is synthesized by a template-directed incorporation of deoxynucleotide monophosphates into the 3' end of the DNA chain. What does that mean? Oh my gosh. You have to copy something to make DNA, right? You have to copy DNA to make DNA. So here in black is a DNA strand, and here's a primer base paired to it. And we're adding bases to the 3' end by saying what's on this template, and we'll put in a base that's complementary to it, right? If there's a T, you put in an A. If there's a G, you put in a C. DNA synthesis is what we call semi-conservative. That means both strands are copied. Okay, here's a double-stranded DNA here. Both strands are copied. That's opposed to conservative replication, where only one strand is copied. So here we take one of the strands in this duplex and make it double-stranded, and now we have more double-stranded DNA. The place where DNA synthesis begins is called an origin of replication. We'll talk a little about them. And of course, to make DNA, you need DNA-dependent DNA polymerase and some other proteins as well. And most polymerases need a primer here to get things going, but some don't. Some are primer independent. So here's the structure of a DNA polymerase here in red. And below it is the poliovirus RNA polymerase, and they look similar, which shows you that all the nucleic acid polymerases are evolved from a common ancestor. The polio polymerase is closed. The active site, which is shown by these yellow beta strands, is closed, whereas the DNA polymerase is open. Here's the active site here in yellow. Now, the active site is where the bases are added. Let's get deep into some chemistry just for a moment, okay? Bear with me. The synthesis of DNA requires two metals bound to these beta strands in the active site. And the metals are magnesium typically, and they're held in place by aspartate amino acids. Two of them here, aspartate A and C. And you see that they are holding the magnesium in place. They're in the active site, and those magnesiums are critical for adding bases to the growing strand. So here's the template. We have our sugars and our phosphate backbone, and we have our bases. We've added a C, an A, and a T, and now the T is just being added. It's complementary to the A, and then the next one will be an A. But the T comes in as a sugar with 3 phosphates on it. 1, 2, 3. And we get rid of the last 2, and the energy that we get from breaking this last bond is used to connect the phosphate, the first phosphate, to the ribose. And these magnesiums play a role in that. That's what these little dots are in the red arrow. So just know that in the active site, magnesiums held in place by 2 aspartates help the phosphates make a new bond in the growing chain. Now, the host cell, depending on the virus, provides something. DNA replication always needs at least one viral protein, and sometimes many, to be made, and hence DNA synthesis is always delayed after infection. And viruses with small genomes require more host proteins, so SV40 has one viral protein involved in DNA replication. Viruses with larger genomes encode everything needed for DNA synthesis and even the production of NTP substrates. But they still have to make these proteins to make DNA. So DNA synthesis is always delayed. It's always a late event in infected cells. So where does the polymerase come from? The small viruses don't encode a polymerase. The genome is too small. Like the papillomas, the polyomas, the parvoviruses, they encode proteins that we say orchestrate the host. What does that mean? It means it distracts the host into copying their genome, the viral genome, rather than the host. It's very tricky. We'll see how that works. Large DNA viruses encode most of the replication systems, the herpes, adeno, and pox viruses. In fact, pox viruses replicate in the cytoplasm, so they have no access to the nuclear DNA synthesis machinery. They have to make everything on their own. But adeno and herpes replicate in the nucleus. Nevertheless, they make almost everything that they need to replicate DNA. Besides the polymerase, what do you need? You need DNA polymerase and accessory proteins, an origin binding protein, a helicase to unwind the double strand. You need exonucleases to correct errors, because every polymerase is error prone. And you need enzymes to make the precursors, the dNTPs. All right. Now, the interesting thing about viral DNAs is that the structures are all over the place. They're really different. Polymerase to be or not to be. I like that. Good name for a podcast. So we have single-stranded DNAs, we have circular double-stranded, and then we have a bunch of linear double-stranded DNA. So how are these replicated? Are there any differences? Okay, we're going to go through this. There are 2 kinds of DNA synthesis, all right? Only 2 kinds. There's the replication fork, and there is strand displacement. So these are DNA molecules. The blue of the 2 shades is a plus and minus strand. And a replication fork is just that. The DNA is copied on both strands, and it pulls the 2 strands apart, and it looks like a fork. You know the old saying, if you come to a fork, take it. Well, that's what's going on here. This kind of replication is found in papillomas, polyomas, herpes, and proviruses of retroviruses. This is how our DNA replicates, by a replication fork. And it needs RNA primers. These green areas are RNA primers, and that leads to an issue that we'll see in a moment. The other is strand displacement, where basically the one strand is copied and the other one is just pushed off. That's why it's called displacement. It's not a replication fork because both strands are not being copied. This happens with adenoviruses or parvoviruses. This is never RNA primed. The primer is either a protein or a DNA. It's a protein for adenoviruses and a DNA for parvoviruses. Okay? Now, the problem in DNA synthesis is the end, the 5' end problem. And here's what it is. If you have a linear double-stranded DNA, like our chromosomes, linear double-stranded DNA, then you separate the 2 strands because you're going to copy both. You make an RNA primer and then use that to make DNA, which is shown red here and orange here. And then you don't want RNA in here. You want DNA, right? So you remove the RNA, but now you're stuck. You have a gap, which you can't fill in because to fill it in, you need an RNA primer. And then when you take the RNA primer out, there's a gap. So that's the 5' end problem. And in our chromosomes, it's solved by having telomeres, which are very, very long repeats. They slowly get shorter and shorter and shorter because of this end problem. And that's how we age, by our chromosomes getting shorter. And by the time you're very old, they're very short and they're starting to chew into genes over there, and it's a real problem, right? Telomeres, okay? How do viruses solve? Viruses can't do anything, but how is the 5' end problem dealt with for viruses? We're gonna talk about that for a couple of different viruses. All right, so SV40 was the first DNA replication system of any kind because people couldn't figure out how to study DNA replication in eukaryotic cells. The DNA was huge, but SV40 is 5,000 base double-stranded circle, very easy to purify. You just purify virus particles and you have DNA, which you can then add replication extract and get it to copy. So what they found was when there's an origin of replication in SV40 DNA, that's where DNA synthesis starts. And in fact, it goes in both directions from the origin. And so here's our DNA. Then we have melting of the DNA here, and the replication apparatus comes in and begins to copy both strands. And here is our replication fork. So both the top and the bottom strand are being copied at the same time. All right? We know this happens. We have experimental data that says it's bidirectional. Semi-discontinuous DNA synthesis from a bidirectional origin. A lot of stuff packed in there. So here's the origin. Bidirectional means the DNA synthesis occurs in both directions, on both strands. Semi-discontinuous. I was thinking about this morning on the train. It could have been semi-continuous also, right? Because what it means is that one strand can be continuously copied. You put an RNA primer there, and the polymerase uses it as a primer and just keeps copying. And as this replication fork gets bigger, you get more and more DNA. And that happens on the other strand as well. These are called leading strands because the polymerase latches on and just goes. But the other strand, you can't do that because it's the wrong direction, right? So look at this fork here. You can't copy any further than the fork has opened. You have to put primers down. So as the fork gets bigger, you put this primer down first. It fills it in 5 to 3'. You can't go in the other direction. That's not allowed chemically. Then as the fork gets bigger, you put the second primer in, third, fourth, fifth. You end up with all these short DNA fragments with RNA primers. That's called the lagging strand. And that is discontinuous, right? You have to make each piece at a time, whereas the other strand was continuous. So that's why it's semi-discontinuous or maybe semi-continuous. I don't know. But there's no end problem here. Can you see? Well, because there's no end. There is no end where you're going to have a primer left. When this synthesis has gone all around the circle, you just remove the primers and you use the DNA to prime synthesis to fill in the gap. And that's it. So the end problem is solved by making the genome a circle. Here's how it works, though. This looks complicated, but I'll simplify it for you. So here's SV40 DNA. There's the origin. And this DNA goes in the nucleus, and it needs the cell to replicate it. How does it do that? How does it say, come here and replicate me? It's large T antigen. T antigen is the first protein made in SV40 infected cells. And DNA synthesis doesn't happen until T antigen is made. And 6 of these bind on either side of the origin. What do? First of all, they denature the origin. They separate the two strands. And they bring in this cell protein called RPA, which binds to the single strands and keeps it single-stranded. And T recruits RPA. And finally, the third function of T, it continues to melt or displace the two strands, denature the two strands of DNA. So DNA, large T is responsible for recruiting the cellular DNA synthesis apparatus to the SV40 origin. And this is the bubble, the bidirectional bubble we just saw previously. It just gets bigger and bigger and bigger until it goes all the way around on the circle. And now you have two circles. However, it gets bigger and bigger. As you can see, the RNA primers are removed. It's filled in with DNA, and you have two continuous DNA strands with no end problem. But the problem is at the end, the two circles of DNA, they both have new strands, right? So if you've gone from 1 to 2, they're linked. They're entwined into each other, and they can't separate. So what do you do? Well, there's a cell protein called topoisomerase 2, which cuts one of the strands and allows them to separate. And then the cut is resealed. I think this is just so brilliant, right? Because you got this twisty thing. There's an enzyme there. Cut it, and you're all set. Also, during replication, when you have this this bidirectional fork, that denaturation of the fork twists the rest of the molecule and makes it overwound. And that periodically has to be relaxed. And another, again, topoisomerase, it cuts one strand and that relaxes the supercoil. You can imagine a highly twisted rubber band. You make a little nick and boom, it all explodes again here. These topos are great. I love them. Okay, so that's SV40. Parvoviruses are single-stranded DNAs. They have a built-in primer, which is their own genome, because the ends are folded and there's a primer. So here's how the DNA folds on itself, and that makes a perfect primer for DNA synthesis. And so the way this works, there's no end problem. This is a bit complicated, but let me show you part of it. Here's the viral genome in its hairpin formation. The cell then fills in the top strand, makes it fully duplex, as you can see here. A viral enzyme nicks near this hairpin, and that allows both ends to be fully synthesized. So now you have two DNAs, and there's no end problem because there was never an RNA problem. And so you don't have to remove the RNA. And these two strands are separated. There's strand displacement to copy this strand and pop off the old one. So in the end, you get multiple DNA molecules from one. But it's DNA priming, so there's no end problem. For adenovirus, it's protein priming. The DNA is linear double-stranded DNA, and the polymerase binds to each end. In fact, the primer for adenovirus is a protein. Here is the end of the DNA. The double strands have been separated. The polymerase comes in with a protein linked to a C, and that acts as a primer to start copying the genome. And so you prime with the protein, so there's no RNA to remove. End problem solved. You use a protein as a primer. And so this is strand displacement. You copy one strand, make a duplex, then the displaced strand is copied again. Semi-conservative replication. And finally, herpes simplex virus encodes all the proteins it needs to reproduce its genome. And so it's independent of the cell. The DNA enters the cell as a linear molecule. It's converted to a circle in the nucleus, and then it replicates by what we call rolling circle replication. So the double-stranded circle is nicked. The 3' end of the DNA is used to start DNA synthesis, and that keeps going around the circle, and the new double strands are coming off. One of my students said, it's like toilet paper, right, Professor? Each sheet is a genome, right? That's the way it works. There's no end problem because the RNA primers are removed on this discontinuous strand, and the DNA in front of it just serves as a primer to fill in the gap. Rolling circle replication. Poxvirus DNAs replicate in the cytoplasm, and they encode all the proteins needed for DNA replication. And the poxvirus genome is weird. It's double-stranded, but the ends are covalently linked. And so these viruses replicate in the cytosol. So here's an infected cell, and in the middle we've stained for a viral protein, the DNA binding protein, and it's in the cytoplasm. The nucleus is marked by DAPI stain, which is blue. And you can see the DNA binding protein is in the cytoplasm, not in the nucleus. So the genome is replicated by RNA primers. You start with a dimer, you start with a monomer, which is all base paired. The replication begins at both ends. You end up with a dimer, and then the dimer resolves into two monomers, which are basically two genomes. There's no end problem because these RNA primers are removed, and the gap can be filled in by the DNA that's in front of it. Okay, it's a brief overview. I want to tell you one more thing, which is really important because this has to do with how viruses cause cancers, which we'll talk about another time. Most of our cells in our body don't divide, right? Did you know that? Your epithelial cells, your skin cells, your mucosal epithelial cells, they're dividing often, but most others do not. And the problem is that viruses don't replicate well in quiescent cells. A quiescent cell doesn't replicate. So what happens when viruses get into cells? They make the cells synthesize DNA replication proteins because that's what they need. The division of the cell is associated with making DNA polymerase and all that, and that's what the virus wants. So the smaller viruses, the SV40, papilloma, and adenovirus, they encode proteins that make cells divide, right, so that they can replicate. So here's cell division, right? You have a phase, a 24-hour cycle where the cells divide, but then after they divide, there's cell growth, there's a period of DNA replication, and then they're big and double DNA and they divide, okay? This division is tightly controlled. I just said not all your cells are dividing. And it's a protein called the retinoblastoma protein that is a checkpoint for cell division. It controls entry into S, which is when DNA is made. This was discovered because kids who have lost the gene for RB get retinal tumors early in their life. So it tells you something right away. If you take out this gene, the cells keep replicating, and that leads to cancer. So how do viruses make cells divide and therefore make DNA polymerase? They encode early proteins like SV40T, papillomavirus E6 or 7, and adeno E1A. Here they are, these purple proteins here. And they bind RB, right? That checkpoint protein. They bind it and prevent it from being a checkpoint. And so the cells keep dividing. How they do it is a little complicated, but I'll explain it. Normally, RB is bound to a couple of cell proteins that are needed to transcribe the genes encoding cellular replication proteins. So if RB is present, these genes are not going to be expressed and the cells are not going to divide. If a large T or E7 or E1A binds RB, now the promoter can be turned on. The cell makes all the genes it needs to divide, and the virus can use those as well. So this is called abrogation of RB by viral proteins. It's going to be a big player in viral cancers, which we'll talk about later. I think we already talked about it once, but it's time to do it again. And there you go. Mini lecture. You have to make DNA, and nothing is boring, okay? Nothing is boring. Yeah, virus is a Latin word for poison. We used to think they were liquids, but when we saw them in the electron microscope, we realized they weren't. Tona Rona wants to know How accurate are the tests? They're accurate. They're very accurate. They have low false positivity and high sensitivity. Thank you for your contribution, John, to science communication. We appreciate it. Oh, look who's here, Silvio Pina. Thank you for your support. Good to be back in LA. I was in Italy and got a med for 36 euros. Same sells here for 500. I may be leaving the U.S. sooner than I thought. It's all too much. Oh, that's too bad, Silvio. But yeah, I understand. Italy is very nice. Carol listened to Immune 26 with Michael Minna discussing immune amnesia. Vincent said he was looking forward to 2020. I wonder why I said that. Yeah, we mentioned this mandrake, the outbreak in the Congo. I don't know what it's about. I don't know if the bats are really actually part of it or what. It's not Marburg. It's not Ebola. I don't know what it is. Will says, I spent my whole life living and working with the most wonderful Americans. The orange regime is shocking. Well, Will, I agree with you. I think it will reverse at some point. I just don't know when that's going to be. Tona says, most of those saying religious exemption are prevaricating an excuse. So prevaricating means, talking in a way to evade the truth. It's a lie. I use prevaricate as lie as well. So that could be right. Yes, I didn't get a chickenpox vaccine either. I got chickenpox though. Abdul Rahman says shakshuka is a North African dish with eggs cooked, tomato sauce, and possibly beans. Very hearty but light. Sounds good. Logotic says it's you can get it at the Mirador Cafe in New York City. I wonder how far away is lab-grown chicken tissue. Probably not too far. If people will have it, though, right? Hello, Sabrina. I'm disgusted by a plethora of disasters. Yeah, it is disgusting. Inkpot, thank you for your contribution to science communication. I never quite understood the difference between Nipah and Hendra viruses in Paramyxoviridae. So they're both, they cause encephalitis. Hendra was originally, isolated in Australia, in Hendra, the town of Hendra. And it was an outbreak in racehorses and their trainers. The virus went from fruit bats to the horses and then to the trainers. And then Nipah emerged in Malaysia, and it went from fruit bats to pigs to people. So they're just slightly different viruses. In fact, the glycoprotein of Hendra will actually protect against Nipah infection. That vaccine is being tested as a vaccine for both. So they're very close, but they arose in different parts of the world. What I find interesting is that, not interesting, but in Malaysia they had to cull 1.5 million pigs because they didn't want the infection to spread. Can you imagine 1.5 million pigs? Oh my gosh. They just dug big pits and pushed them in. You can find YouTube videos of that. I find it so sad. The poor animals. Oh dear. Sabrina is vegan. I feel for the birds at the hands of humans. I'm disgusted by people. Humans are the worst species doing the most damage. Well, we do. We're screwing up the earth, for sure. But yeah, the consumption of animals is problematic. I feel badly for the animals that are slaughtered and consumed. People are trying to make meat alternatives. There is an ecological consequence to raising so many pigs, for example. And I don't know where that's going to head because many people just like their meat. But I find the meat substitutes actually fine. Yeah, the miasma of anti-think has returned for sure. Sabrina says people need to go out and protest and do what they do in Europe. The government is to work for their people, not against them. Oh, hallelujah. And it did work for us for many years. And now these guys got control of it because that's the weakness of a democracy. You scare people and you lie to them into thinking they're issues. Yeah. German measles is rubella virus and measles is measles virus. Yeah. Here's a link from Vanity for the miasma shirt. Countering a miasma of anti-think. Oh my gosh. Jeff, is there a worry that YouTube will ban you? Nothing happened. People have been asking me if I were banned, what would my plan be so people could get in touch with me and find my content? Because I would continue to make content. I would just post it elsewhere. So, but no, there's no threat. It's just that people have been writing to me, and so I thought I would share it with you. Yes. I demand you provide me with a men's T-shirt. Come buy one at Cafe Press. And we get $1 on each item. It supports us. Yep. I hope you're having a good day. I'm thankful for your published postings of virology. My institution doesn't offer it. I'm glad we're able to help. Is there an oral measles vaccine? Nope. It's just an injected infectious attenuated vaccine given with measles, mumps, rubella together, 3 different viruses. I was wondering if there are any courses I should try to learn before I listen to my virology course. I think just the basic biology course is fine, but I think I explained things pretty well. So, you don't need to really do anything. Yeah, this is very interesting. U.S. military does extensive vaccination, so I have a feeling that the military is going to do its own thing in this country. It'll be interesting to see. Rita says we've been breeding sheep to be resistant to prions. Most sheep are resistant. So how do you breed them to be resistant to prions? You start with a deletion of prion gene or what? Why does a misfolded protein present so much danger? Because when it gets in you, it causes your normally folded proteins to misfold. And it causes a lot of them to misfold, and that causes a pathology, which is the prion disease. That's a short answer, but it's got this weird property. Once it's misfolded, it wants everyone else to be misfolded as well. Every other copy of its same protein, yeah. This is a good point. I think people believe that being infected is synonymous with being sick. I think you're right. I think that most people feel about it that way. Yes, yes. Can a vaccinated person who's infected with measles but doesn't get disease infect another? I think that the capacity to transmit is greatly diminished. It's not zero, but it's greatly diminished. Yeah. I'm looking through the comments because we got to wrap it up here and seeing what else we have. You guys are chatting away during my lecture. It's totally fine. I don't mind. It gives you a chance to speak amongst yourselves. Yeah. Artemis says TWIV requires a fair amount of background knowledge to fully follow. Yeah, it's true. Your lectures are hard, but your clinical update and office hours are understandable. What about TWIV? You find that very hard? Markle, nice to see you. Stay safe. If a hen is infected with bird flu, not showing signs, could the virus be passed into the egg? No. No, the virus doesn't pass into the egg, as far as I know. The reason there's an egg shortage is because they cull the herds that are infected. So now you have no more layers, right? You guys have a great time here. There are lots of great chats and comments. Oh, Eliza says, Mom is a scientist and she loves the bullet points. Very good. Surely everybody at CDC will tell him he's wrong. I'm not sure. They want to keep their jobs, right? That's the problem. He just fires people. You know, this nonsense from Musk. He sent out an email to all the federal employees. What did you do last week? And if you don't answer, you get fired. What kind of BS is that? We're grown-ups in this country. This is just nonsense. Stop it. Aye. Yeah, regarding phages, remember their goal is to make more phages, not to protect humans from bacteria. That's true. We are repurposing them. How common are adjuvants in vaccines? Common in Europe, not so common in the US, but there are some. For example, Shingrix is adjuvanted, and some others which I can't remember right now. Bree Star, thank you for your contribution to science communication. Really appreciate it. Questella, thank you for your contribution to science communication. I'm sorry I have to skip through all the other questions. This one crazy. Thank you for your contribution to science communication. This is why I don't want to do a mini lecture because I want to read all your questions. Wow, you guys are amazing. I'm actually very impressed that you, yeah, I love a mini lecture, but responses to comments are also nice. But a bunch of people wanted to do a mini lecture, but maybe next week I won't do a mini lecture. Questella says, your lectures are like beating the system. A school I could have never gotten into, never been accepted into, but now I'm here and I don't know. Thank you. Yeah, you're totally welcome here. Absolutely, everyone's welcome. All right, I think I have to stop answering questions and just see if there's anyone to thank for donations here. Damn, such good comments. I like this one. I can't see Twiv being shut down. You're blinding me with science. Oh, yeah, that's great. Your mini lectures take up one notebook page, front and back. All right, let's see. Go to the bottom. There you go. I want to thank our moderators tonight. Let me get rid of this comment here. We don't need that up anymore. Thanks to the moderators who were here tonight. I know that. Let's go away to the beginning. We had Les. We had Barb Mac UK. We had Vanity Nutrition. We had Tom Steinberg. Thank you so much for moderating. I did say Vanity, right? And I said Les. Thank you for moderating. I know you were very busy tonight. Sorry about that, but we're attracting more and more people, and that's what's going to happen. So I appreciate your work. I appreciate everyone else coming and continuing the chat while I'm mini lecturing. Lots of good comments here. Wish I could get to them all. When are we going to be back? Let's take a look at the calendar. Today is the 26th. Next Wednesday is March 5th. Yeah, March 5th. We're going to be back at 8 p.m. Eastern for another office hours. I promised Questella I'm going to stream to Twitch. At the same time, I will bring pictures of Dickson's photographs, and I will and no mini lecture next week, just chatting with you guys, all right? Even though you like mini lectures. I have plenty more planned for you, but I want to get through all those chats. I like it. It's a lot of fun, right? Okay, folks. I hope to see you next week. In the meantime, I want you to take care of yourself and write your representatives and senators every week and complain about what's going on, all right? Take care, everybody.